right, guys. Again, thank you so much for accommodating me the other day on Zoom, even though doing Zoom lectures is not exactly ideal, but at least we were able to get through the majority of the important information in part one. There are a couple things today that I want to just briefly do before we finish up with part one of chapter one. I did want to play some of these videos since we weren't able to really do any of the videos over the Zoom lecture. So first, let's watch the video on genetics and genomics, so splicing or splice variants. So let's go ahead and click on that.
In many genes, there are parts of DNA that are not used to make the protein. These unused or non-coding areas of the gene are called introns. The portions of the gene that have the instructions for proteins are called the coding portions or exons. When DNA is used as a template to make RNA, both exons and introns are included in the RNA. Before that RNA can be used as a template to make the protein, the introns must be removed. The process of removing introns from the RNA sequence is called splicing. There are specific places between the exon and introns called splice sites that allow our cells to remove the introns. These splice sites are represented by scissors. Once the introns are spliced out or removed from the RNA, the remaining exons join together to form the final RNA product. These remaining codon sections are then used to create the protein. We all have changes in our DNA that are called variants. Some variants do not cause any health concerns, but other variants can cause disease. Some genetic variants can affect the splice sites that allow our cells to recognize the introns and remove them before creating a protein. A variant at a splice site or that affects splicing might be called a splice site variant. Variants that affect splicing can result in the loss of exons or the inclusion of introns. The loss of exons or inclusion of typically non-coding introns can change the protein that is made. In this example, there is a splice site variant in the gene. The variant changes the letter G to a T. This variant is located in the splice site where the cell recognizes and removes the introns from the exons. This variant causes the cell to no longer be able to recognize the splice site, leading to the removal of an exon. Removing an exon then leads to a shortened protein. This is represented by the red dots with the X, indicating that part of the protein should be present, but because of this variant is missing. But when a protein is shortened, it can impact how the protein functions in the body. Thank you for watching our splice site variant video. If you have questions, please reach out to your healthcare provider or contact Genome Connect.
All right, cool. Let's move on.
I have a quick question.
Yes.
So I don't know anything about genetics.
Yeah,
Is it? So the beginning is the intron and the exon, that's DNA, and then DNA does the process of the splicing that we're trying to copy to make RNA?
there's going to be all sorts of different things within the introns that actually contribute to whether or not they're going to be sliced at different areas, right? And so people call introns junk DNA, but that's actually kind of a misnomer. There's a lot of information still packed into the introns that actually do contribute to what like your body naturally does when it comes to actually rearranging the exons. And so there's going to be things found within the introns that help to actually do that, right? There are going to be like things like transcription factors, things that help to rearrange exons. Right? So it's all encoded within the DNA. I don't know if that answers your question.
Yeah, I just got to learn more about that.
Yeah, no sweat. Again, this is not I'm trying not to make this a genetics class, but it's kind of turning into it because there's a lot of genes that are going to be involved in, and as well as transcription factors and things like that involved in neuronal signaling. So, and then the other one that I wanted to play as well, these guys over here. So we talked about, this is basic genetics, right? Dominant versus recessive alleles, the difference between genotype versus phenotype, things that you should have already learned in other classes, basic biology. But let's watch something now that talks a little bit about homozygosity mapping. And this can help to try to identify disorders, genes that cause disorders.
Welcome to Gene Clips. My name is Gabrielle, and I'm a genetic counselor at Children's National Hospital. Today, I will be talking about areas of homozygosity. Sometimes this is also described as runs of homozygosity or loss of heterozygosity. To quickly review, we usually have 46 chromosomes in each cell. We receive one set of 23 chromosomes from our father and one set of 23 chromosomes from our mother. Areas of homozygosity are regions of chromosome pairs where the genetic information of both copies, the one from the mother and the one from the father, are identical. Most often, areas of homozygosity occur when parents are related to one another, even distantly. The size and number of identical regions depends on how closely related the parents are to one another. Areas of homozygosity are often identified by the genetic test called a chromosome microarray. A specific diagnosis is not made by this result and may not affect a child's health. However, these shared areas sometimes result in an increased chance of having a genetic condition. For this reason, additional genetic testing may be recommended. Your genetics provider is available to review this information and the significance of the result for your family. Thank you for watching Gene Clips.
So why is that clinically relevant? First of all, just because a person is maybe slightly related doesn't necessarily mean that it's incest, right? So one thing that you might see in certain populations, for example, like Ashkenazi Jewish populations, when like a Ashkenazi male and an Ashkenazi female are thinking about having a family together, it's often recommended that they do genetic counseling, right? Because within Ashkenazi Jewish populations, you have certain conditions that might be, in higher rates within those populations. There's, like, for example, Tay-Sachs disease is a good example of a disease that you find within Ashkenazi Jewish populations. And so just to make sure that either the someday dad and the future mom don't have the actual trait, right? Maybe the recessive allele. You want to do genetic counseling to rule that out. So, like one of my colleagues, for example, he's Ashkenazi. His wife isn't, but regardless, they went just to have fun. They went to get genetic counseling, and he actually is a carrier for a couple traits of diseases. I forget which ones off the top of my head, but if his wife was indeed Ashkenazi as well, and was also a carrier of certain alleles that might, result in things like Tay-Sachs, they would maybe want to at least prepare themselves in having kids, right? For the chance that there might be like a recessive trait within their offspring. But then, if the closer a person is related, right? I'm talking like cousins, siblings, even cases of like true incest, right? That's why at least in like biological terms, genetic terms, like incest is a bad idea because you're going to have an increased rate of recessive traits coming through in the offspring. So homozygosity mapping can help basically identify the types of diseases that might be at least phenotypically represented if they had the genotype for those recessive traits. And here's another one I wanted to play, genome-wide association studies. And so another way that you can try to study for different types of genetic disorders.
Phenotyping algorithm is very important in supporting genome-wide association study. What is a genome-wide association study? It's an approach that involves scanning biomarkers such as single nucleotide polymorphism, or SNPs, from DNA of many people in order to find genetic variation associated with a particular disease field phenotypes. Once new genetic associations are identified, researchers can use that information to develop better strategies to detect and treat and prevent diseases. So, how are genomic-wide association studies conducted? To run a genomic-wide association study, or GWAS, we first identify the disease phenotypes. Then group the participants into these two groups, cases, and these are people with disease phenotypes. And controls, those are similar patient without the disease phenotype. Then we need to obtain DNA samples from all these participants. From DNA samples, we can use lab machines to quickly survey each participant's genomes for genetic variation, which are called single nucleotide polymorphism, or SNP. If certain genetic variation have found to be significantly more frequent in the people with the disease phenotypes compared to people without the disease phenotype, these variations are said to be associated with the disease. We do this by computing the frequencies of SNPs on the cases and on the controls. Then, based on the frequency, we calculate the odds ratio. From there, we can calculate the corresponding p-value for the odds ratio. If the p-value is small, then we conclude this variation is significant. The associated genetic variations can serve as powerful pointers to the region of the human genome that may cause the disease. Here's an example showing more details on how the GWAS is computed. We first identified the cases and controls. That is, the people with the disease phenotype and the people without the disease phenotype. In this case, we have 4,000 patients with the disease phenotype and 6,000 patients without the disease phenotype. Then we iterate over all the SNPs to compare the relevant frequencies. For instance, for SNP1 for the control group, we have 2,676 out of 6,000 has the corresponding variation G at this location. And the frequency of G in this case is 44.6%. In the case group, we have 2,104 out of 4,000 with the corresponding variation G at this location. So the frequency is 52.6%. If we go through the calculation, we'll find now the p-value is 5 times 10 to the minus 15.
All right, now it's getting a little bit too heavy into the statistics of it. But another thing that he didn't mention, he was associating like single nucleotide polymorphisms with disease. Not always the case. You can also have single nucleotide polymorphisms that could confer certain benefits to individuals. I remember back in the height of the COVID-19 thing that was going on. There were some studies that were actually looking at individuals of certain demographics that had lesser rates or lesser severity of the viral infection. And for those individuals, it had to do with single nucleotide polymorphisms in those populations. And those actually conferred benefit to those individuals. So it can work in both ways. Single nucleotide polymorphisms could result in disorders, right? Or sometimes, depending on the condition, it could also result in benefit for the individuals. So make sure you know your transgenic animals. Make sure you know your knock-in or knock-out animals. Again, with transgenic animals, that means you're taking DNA from one type of animal and you're basically integrating it into the genome of a new animal, right? So for example, with these little mice over here, they were able to get some green fluorescent protein so they could light up underneath a black light. Again, one of my RDP that we're putting together, right now we're doing cell models, but eventually we're going to be doing rodent models. And the rodent models that we're going to be using are alpha-synuclein transgenic mice. And so they have a SNCA mutation, and that's actually derived from human SNCA, alpha-synuclein mutations, and they were integrating that into those transgenic mice. And so you can induce Parkinsonism on those types of transgenic mice much more readily than you would other types of rodents, for example. And so that's kind of the idea behind a transgenic animal model. Knock-in and knock-out animals, that's where you insert or remove a gene and see what kind of effect that's going to have on the individual organism. And this is where we left off last time we met. So we were going to discuss homologous recombination as well as CRISPR-Cas9. Homologous recombination, I'm not really going to hold you guys accountable for this, but let's still watch a video on it. Just, so you guys can be exposed to different techniques. But I will likely include CRISPR-Cas9 as part of your upcoming exam. So we'll watch a video on that as well.
Homologous recombination underlies many genome engineering methods. It involves inserting DNAs into the genome at arbitrarily chosen locations based on their homology to that local sequence. In E. coli and other bacteria that lack efficient homologous recombination systems, this functionality can be enhanced by expression of lambda-red recombinase. Lambda-red is in common use for producing knock-ins and knockouts in prokaryotic genomes. Mechanistically, the recombinogenic species is a linear single-stranded DNA, such as oligonucleotides, that is introduced into the cell by transformation. Alternatively, the linear double-stranded DNA can be provided, and the exoprotein of lambda-red will degrade it into single-stranded molecules that can undergo recombination. Often that linear DNA is a PCR product. The method does not work directly on circular DNAs, but linear DNAs can be generated from circular DNAs by treatment with any of the various restriction enzymes. A very popular setup for homologous recombination in E. coli and other prokaryotes is the Dotsenko-Wanner method. It is a lambda-red-based method for knocking out genes in the genome. It begins with pretty much any E. coli string containing the target sequence we wish to remove. The cell is first transformed with a helper plasmid called PKD46, and it's grown at the permissive temperature of 30 degrees. This temperature-sensitive plasmid encodes the lambda red genes under a PBAD promoter. Growth of the cells with the rhabinose present will induce expression of this cassette, resulting in the production of lambda red and the enhancement of homologous recombination in the cell. The knockout cassette begins with a template plasmid. The plasmids PKD3, PKD4, and PKD13 were originally designed for this experiment, and they essentially are sources of the chloramphenicol resistance gene for PKD3 or the kanamycin resistance gene for PKD4 or PKD13. There are specific 20 base pair regions of the plasmid called P1 and P2, which are the sites where oligos can prime to amplify the selectable marker in PCR. These PCR oligos are designed to contain these 20 base pair sequences on their 3' ends, and then 40 base pairs of homology to the genome target on their 5' ends. PCR results in a double-stranded linear PCR product with homology to the genome on both ends. The cells containing the lambda red genes are transformed with this PCR product, usually by electroporation. Inside the cell, the lambda red genes cause the double crossover recombination of the PCR product over the sequence homologous to the ends in the target. Because recombined cells contain the chloramphenicol resistance gene, they can be selected by growth on antibiotic-containing medium.
So that, anyways, I'm going to end it there. That gives you a good idea of kind of what's going on with homologous recombination technique. So basically, he was creating more and more and more of that genetic sequence using PCR. You guys know how PCR works, right? So polymerase chain reaction, you just basically amplify the strands of DNA that you're looking for, and then basically transforming the bacteria to introduce that strand of DNA into the target region. Now, the other one that we're going to talk about is CRISPR-Cas9, and that's also another way to basically introduce mutations into the genome of an organism. And so this is going to be one of the major ways that you undergo gene editing. So CRISPR-Cas9, pretty novel stuff. It's gotten really popular over the past 10, 15 years or so. And yeah, super cool. So let's go ahead and watch this video. I think we'll watch the entirety of it, all 4 minutes.
Every cell in our body contains a copy of our genome, over 20,000 genes, 3 billion letters of DNA. DNA consists of 2 strands twisted into a double helix and held together by a simple pairing rule. A pairs with T and G pairs with C. Our genes shape who we are as individuals and as a species. Genes also have profound effects on health, and thanks to advances in DNA sequencing, researchers have identified thousands of genes that affect our risk of disease. To understand how genes work, researchers need ways to control them. Changing genes in living cells is not easy, but recently a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The CRISPR method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces 2 types of short RNA, one of which contains a sequence that matches that of the invading virus. These 2 RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus. Over the past few years, researchers studying this system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAM. The Cas9 will unzip the DNA and match it to its target RNA. If the match is complete, the Cas9 will use 2 tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut. But the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise, for example, by replacing a mutant gene with a healthy copy. This can be done by adding another piece of DNA that carries the desired sequence. Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version. All this can be done in cultured cells, including stem cells that can give rise to many different cell types. It can also be done in a fertilized egg, allowing the creation of transgenic animals with targeted mutations. And unlike previous methods, CRISPR can be used to target many genes at once, a big advantage for studying complex human diseases that are caused not by a single mutation, but by many genes acting together. These methods are being improved rapidly and will have many applications in basic research, in drug development, in agriculture, and perhaps eventually for treating human patients with genetic disease.
All right, so definitely great stuff. Now, what kind of issues do you foresee with those types of gene editing technologies? Yeah, what kind of ethical issue might come into question?
Playing God.
Playing God. What else?
It could be used for too.
Oh yeah, definitely, absolutely. So, since there's a lot of people who are concerned about like synthetic biology, for example, where you can actually we're like probably about 5 to 10 years away from basically kind of like 3D printing, just starting off with a code, right, of like genetic code, and then being able to like 3D print something that's based off of that genetic code.
What about like cloning?
Cloning would definitely be a concern. But with this, like I think personally, what I find most concerning, like playing God, right, but also eugenics, right? So like eugenics, designer babies, right? We can already do that. We can already select for certain traits, now, and even like people who are doing like surrogacy and like, buying children and stuff. Even that's becoming a huge controversy nowadays because it's like, yeah, designer babies,
Brave New World.
Huh?
Brave New World.
Yeah, it's basically Brave New World. But like CRISPR-Cas9, that's kind of one of the major concerns. So like the bioethics behind technologies like that it's no joke. We as a culture and as a community, especially if we're going to work in the scientific community, these are things that are going to be coming up, right? Being able to choose how tall someone is, how smart they are, how able they are when it comes to like physical abilities and things like that. So huge concern, but also the promise to get rid of certain diseases is also a really, potentially beneficial thing that you can see with this, like for example, Huntington's disease, horrible condition. When somebody gets Huntington's disease, it's from no fault of their own. It's, they have a trinucleotide repeat, CAG repeats. The more of these repeats you get in your genome, the earlier on in your life you will develop Huntington's. So they actually have they call a juvenile onset, even though it's still not young. It's like a person in their 30s, basically. But once, if you see your parent getting Huntington's disease, because it's a dominant disease, it's autosomal dominant, your chances of having Huntington's are significantly higher. And then you go get genetic counseling, and then they tell you, yeah, sure, you actually do have the genetic traits. You have the genome for it. And one big area of controversy with genetic counseling for Huntington's disease is the rate of suicidality in people who test positive because they know they're eventually going to develop Huntington's disease. The rates of suicidality is really high in those populations. So those are all concerns. So if you could eliminate Huntington's disease with a technology like CRISPR-Cas9, then that would be great for the people who are affected. But there's gray area, right? There's going to be gray area in terms of the actual bioethics. Anyways, conditional mutagenesis, this is absolutely FYI. I am not going to test you guys on this, but basically this is where you can have basically recombination of your exons. But like I said, I'm not going to be testing on that. CRISPR-Cas9, however, This will be something that you might see on your exam. So look at this quote from one of the co-inventors of the technology. It was one of those oh my god moments when you look at each other and say holy moly, because this is some pretty wild stuff. The things that you're going to be able, that as a scientific community, that we'll be able to do in the coming years is going to be really, really impressive. But then again, we also have to be very cautious, right? Cautious where we go. We don't want to be playing God. So in terms of research, these are all your model organisms, right? Cats can be used as model organisms, rodents, such as this useless creature over here, my wife's guinea pig. So rodents can be used as model organisms. It's not on here, but C. elegans, you guys familiar with C. elegans? It's like a tiny little worm, right? C. elegans, the entire neuronal system of C. elegans was mapped years and years ago, and it's one of those organisms that has actually provided a lot of information about conditions like Parkinson's disease and a lot of other like neurotransmitters and stuff like that. So C. elegans, even though it's a tiny little worm, right, we can still derive a lot of information for when we do research on those types of organisms. Fruit flies, we're going to talk about Drosophila quite a bit, like all the Hox genes and stuff like that. We're going to talk about that when we discuss actual like embryogenic development. And so fruit flies have been used as a model organism to study lots of different areas of genetics that crosses over into human genetics as well. So pretty cool stuff. Anyways, I still love it, even though it's totally useless. Wilbur is his name. He looks like a Wilbur. All right, let's do some quizzes questions before we get into radiology. Radiology is not going to take too long, so I imagine that we're actually going to blast through it and then even begin embryology or development in a little bit. So hopefully we'll begin that by the end of today's class. We'll go ahead and log in. All right, cool. Let's go ahead and begin. All right, so make sure you know the structures of your neurons. So let's see how you guys did. No, you guys did great. That's perfect. So your dendrites, your somas, nodes of Ranvier, the myelin sheaths, the different types of cells that create myelin. We already talked about that. Oligodendrocytes versus Schwann cells. Super softball question. I expect all of you guys have gotten that correct. Beautiful. Most of you guys got that correct. So make sure you know CNS, that's going to be oligodendrocytes. Peripheral nervous system is going to be your Schwann cells. Okay. Oligodendrocytes can myelinate multiple neuron axons, whereas Schwann cells only do a one-to-one. All right, good. Super straightforward. So that's the neuronal doctrine, right? So you have one neuron, basically they act as discrete units, right, to communicate with other neurons. Whereas with the reticular theory, it was kind of the idea that everything worked together. Think of it maybe as like kind of like a heart, right? That would fit more of like a reticular theory model where you have like the cells all basically connected, working as one. Maybe they're all connected to like gap junctions or something like that. Not actually the way the brain works, right? The neuronal doctrine is what took over. That's the main going theory. And it was really only until we could actually see the cells using electron microscopy when this was actually solidified, right? Verified. So electron microscopy was the major way that you were able to prove that the neuron doctrine was true. All right, softball question, convergence versus divergence, super easy. No explaining necessary. All right, good. Microglia. Microglia are the phagocytes of the CNS, right? Those are what gobble up any sort of waste materials, debris, potentially infectious intruders, right? That's all going to be the microglia. Misfolded proteins, etc. All right. So for the sake of this first exam, these are really the only staining methods that I'm going to be holding you guys accountable for. So missile staining versus crystal violet versus Golgi staining versus fluorescent stainings or fluorescent dyes. So make sure you're familiar with those different types of staining methods. So neuropils can also be associated with gray matter. Right, you're going to find neuropils, especially in areas of lots of gray matter. You're going to have lots of your glial processes, lots of synaptic type regions, right? So that's where cells are really coming into contact with each other. So you find this in, for example, like the cerebral cortex. You'll find collections of neuropils. All right. So we kind of went over the myotactic reflex last time we spoke. So let's go over it again. Let's just go over it using this chart. So this would be if you put sensors within different areas. What's going on here? There we go. All right. So A. So when you have the hammer tap, think about this as being your patella, for example. So when you tap the patella tendon, that's going to be the first thing that happens, right? That's going to trigger sensory afferent cells within the muscle, right? And so that's going to be A, and that's when you see a higher firing frequency within those neurons, right? That's what's going on over here. Then B, that's going to be where you have sensory input, nerves that are innervating your extensor muscles are going to be firing as well at a higher rate. And then C, those are going to be your interneurons. And for this specific myotactic reflex, the interneurons here are going to be inhibitory neurons. And so they're going to basically tell the flexor muscles, the neurons that innervate the flexor muscles, to not fire, basically. So it's going to inhibit the ability for your flexor muscles to fire. And so that's why you get extension of the knee. So whenever you do a patella tap, right, it's going to extend your lower extremity. And so that means that your extensor muscles are the ones that are actually getting stimulated. And the flexor muscles, on the other hand, those are the ones that are being inhibited. And they're being inhibited specifically by the interneurons. And what's the purpose of this? It's basically your body's way of maintaining balance under normal circumstances. So like if your patella got hit by whatever, right, this would help you maintain your balance. It's kind of similar also to the cross extensor reflex. We're going to talk about that later on. That's when you like step on a nail or something sharp, or you have some sort of painful stimulus, you immediately retract your foot, right, to get away from that stimulus. But in order to maintain your balance, you have the cross extensor. So it crosses over at your spinal cord and then allows your other leg now to maintain your posture and balance so you don't fall over. Okay, so we're going to talk about those reflexes in more detail later on, but yes.
So C is the
interneuron. Yeah, C is going to be the interneuron, and it's going to basically send inhibitory signals to the flexor muscle. And then D turns off, as you can see here in this image. I keep on clicking on the wrong one. Sorry about that. So D over here, you notice that the signals basically stop. They cease. It's because they're being inhibited. And so this is going to be your actual flexor muscles. So you have inhibition of your flexor muscles. Does that make sense on the chart? Want me to go over that again, or are we good? Good? All right, cool. Got all quiet there for a second. What happened? All right, so optogenetics. Remember when we talked about this the other day, they're able to insert different types of opsins within the cells. And opsins are basically going to be stimulated by different frequencies of light, right? And so if you shine a laser that has a specific frequency of light, you could turn on a specific opsin, and then that would actually allow the cell to be it'll allow for like neuronal depolarization of that specific cell if you're able to target that particular opsin for a frequency of light. And so it's a good way for you to be able to, figure out exactly what parts of the brain do what. So you can trigger or you could stimulate one region of the brain and see what effect that has in other parts of the brain, right? When we were learning about optogenetics, we were talking about parts of the basal ganglia. Right? They would put like little sensors within the striatum and see if you stimulate those areas, what is it going to do to, for example, like the substantia nigra, right? Is it going to stimulate or is it going to inhibit other regions of the brain? So that's one function of optogenetics. There's other things that you can do with optogenetics too. So there's like research on using light, for example, to maybe cure certain types of diseases. So kind of a really cool area of research in neurology or neuroscience. Looks like most of you guys got this one correct, which is perfect. So excellent. So you have both on A, that's going to be lower frequency, right? Because you're not getting as much, you don't see as many spikes as an example. And then B would be higher frequency. And then if you have lower frequency, like fewer spikes, you're not going to get as much stimulation in those neurons. So the higher the frequency, the greater the number of spikes, the more stimulation you can get of that neuron. You had a question?
Okay, so if I remember correctly, the amplitude never changes, just frequency.
Exactly.
So
Yeah.
is that why, like, when we talk about like muscles, how like the
Tetany?
Yeah, like stuff like that, where like
Yeah.
the motor neurons, like if I'm doing a big movement, I'm going to recruit different neurons than if I'm doing a small movement.
Yeah.
Is it because I could never increase the amplitude of the small neurons enough that you have to like switch to the big neurons?
That would make sense. That would make sense. That's one way to look at it for sure. I mean, the other way to look at it as well with muscle contraction is looking at things like trepa, right? Remember, that's where you have like or it goes like stairwise, and then eventually it reaches tetany, right? Like the more frequency, like the higher frequency you have in terms of like neuronal firing will eventually result in tetany. So yeah, that's one way to look at it, where you have recruitment of different muscle groups that serve in different functions, right? I mean, even if you look at your cortical spinal tract, you have your lateral cortical spinal tract, which is going to be more associated with like finer movements, your anterior cortical spinal tract, which is more for like big, large movements that are not as like, defined and well controlled, right? So yeah, I mean, that would be a good way to look at it for sure. Yeah. But yeah, amplitude does not change when it comes to neuronal firing. So if you guys remember this particular study there from your textbook, they were talking about stimulating a receptive field. So they had sensors in place on the cortices of when they were doing a primate study in this particular example. And so they were stimulating certain areas to see if those areas would also get, where it would get stimulated in the brain, in the cortex. And so whenever you have a direct stimulation of a particular spot on your skin of your receptive field, that's going to be like the central receptive field. That's where you actually get the stimulation in the brain. But then everything around that area basically gets dampened down. So you have reduced sensory input coming in from the surround. They call that the surround. And you get reduction in sensory firing or neuronal firing in those regions. So it basically allows your brain to focus on the area that's most important, right? To try to like exclude unnecessary information, basically. And so that's where you get diminished response to a signal, and that would be within the surround. Let's do a few more questions here, and then we'll continue on to radiology. I need to restructure that question because this would be more focused on the central nervous system. So you have nuclei within the central nervous system where you have a collection of neurons basically that operate doing the same type of function. But once you get outside of the central nervous system, when you're in the peripheral nervous system, now we refer to that as ganglia, right? Remember like your dorsal root ganglia, for example, right? So that's part of like your peripheral nervous system. So I would have to probably restructure that question just to specify that. But majority of you guys got that correct anyway. So nuclei. So think about all the nuclei you find within your brainstem that are associated with the different cranial nerves, as an example, right? You have like subthalamic nuclei, right? They're going to be responsible for all sorts of different types of functions. All right, 2 more. All right, another softball question. So introns, non-coding, exons are the ones that actually are coding. Let's do one more question. And then this question is about transgenic animal models. We have crossover from genes from one species inserted into the genes of another species, so that you can, for example, test like Parkinson's and alpha-synuclein transgenic mice. Right? So you're taking one genome or a portion of a genome from one type of organism and inserting that into a different organism. So that wouldn't be knock-in, knock-out. Knock-in, that's where you're inserting certain types of genes. Knock-out, deleting certain types of genes. So I guess knock-in might be kind of sort of falling into like transgenic, but this was more specifically geared towards transgenic animal models. So rock on. Any questions before we move on into neuroimaging and EEG? Cool. All right, let's switch gears then. Let's wrap up with neuroimaging, and then we'll try to begin neurodevelopment after this. So here's a really, really pretty image. That's a MRI, and this was, computerized with all the colors and whatnot. Usually when you do an MRI, you're not going to really see all those colors. That you can do color enhancement later on. Even with CT scans, you can do like color enhancement. Like we have a CT scanner over here on campus. It's actually for rodents. It's really cool. So it's, CT scanner is huge, right? This one is like this big, and it's got like a little tiny tube that you can put rodents in there. So rats, mice, whatever you want to try to image, and you can do it. You can sedate them, so it actually has like you can put in like anesthetic, so you can like keep them sedated while you're doing all sorts of different types of scans. So super cool. I plan on hoping to use that because we bought this machine years ago. We've never really used it for anything. And it's like a $500,000 piece of equipment that's just basically collecting dust and building one. So CT scans are very fun. Same thing with MRIs. MRIs, though, are really expensive. So CT scanner, expensive, but not as expensive as an MRI. You do get better visualization with MRI, though. So we'll talk about all that in a second. So here's an overview of everything that we're going to be discussing. So we're going to be talking about CT, which you can think of CT scanning as basically glorified X-ray. So with X-rays, you're getting just basically one image, right? So it's like taking a photograph, right? That's kind of what you're getting with X-ray. You're just getting one single image, and that's it. You can see a lot with an X-ray, but with a CT scan, you're getting X-ray, but it's in 360, right? So when you go inside the tube, there's going to be an X-ray camera, and you have like the film on one end, and you have the actual machine that delivers or emits all the X-rays, and then you're basically taking one shot, then it shifts, takes another shot, and it shifts, and then you keep on doing that until you get a nice 360 panoramic image of what your you're taking your images of, and it's really, really cool because you can get a lot more definition from a CT scanner. Downside with the CT scan, what would you think before we get into it? What would you think a downside would be? Pretty significant downside. Yes, radiation exposure. X-rays, you get radiation exposure from X-rays, right? Generally, it's pretty safe, right? You're not getting X-rays done all the time. If you work in radiology, you're going to want to wear the garments, right? You'll wear this giant like lead, basically like a vest, and then you also have to wear this goofy little, it's like butterfly type shaped lead garment that you wear around your thyroid gland, because your thyroid is especially prone to radiation. And so you still have to do that if you're working as like a radio tech or if you're working as a radiologist. But generally speaking, you're not getting X-rays done all the time. But if you're getting a CT scan done, you're getting way more radiation exposure. MRI, the only thing that's involved there is magnets, basically. So you're not getting radiation exposure. But there's been certain cases of individuals getting severely harmed because they had metal objects on their body. I'm not going to go into detail in this one case, but there's a class action lawsuit involving a certain recreational item that somebody had within their body, and they didn't tell the X-ray or the MRI tech that they had anything in them. But then when they went through the MRI, there was actually metal within this object, and it went careening through their body, and it caused some serious organ damage. And so, yeah, that's actually a really weird lawsuit. Like I said, I'm not going to go into detail about that specific case. It happened here in Arizona. You can look it up online if you want to get more information about that particular case. But then there was some other really interesting cases, because this is a really powerful magnet. I'm talking extremely powerful. There was two cases. One in Brazil, where a guy, he told the MRI people that he didn't have anything on him, no metal objects. But he actually had a gun on him, and it was loaded, and it was chambered. And so when he went into the MRI machine, as soon as they turned it on, the gun just like discharged, and it shot him in the leg. Actually, no, I think it killed him, if I remember correctly. I think he actually died from that case. So this happened in Brazil. I don't remember the full details of it. But then there was another case that happened here in the States. I think it was a politician, and he had a security guard with him. And so he had to get rushed into the, for whatever, to get an MRI done. And the security guard's gun got sucked out of his, his holster and got stuck onto the machine. Absolutely wild stuff. So anyways, MRIs can potentially be dangerous, but it's only if you have metal objects on you.
If you have metal metal plates.
That's a really great question. Go into an MRI. So titanium is paramagnetic, and it's not going to be affected by the magnetic field of an MRI. And so most of those types of devices, like screws, things like that, plates, rods, whatever, if you broke a bone or something like that, it's usually going to be titanium. And so that is, the risk is extremely low. So you can still get an MRI done. And here are some other more, what would you say? I guess you can see more clarity, more granular information using magnetoencephalography. And then magnetic source imaging basically combines a couple of these different types of techniques together, including magnetoencephalography. Dense diffusion tensor imaging, also known as tractograms. Those are really cool because you can actually color code them, and you can see all the different tracts going from different parts of the brain all the way down into the brainstem, and of course down into the spinal cord as well. So it's super, super cool radio imaging techniques. But anyways, what's the whole purpose of doing radio imaging? I mean, back in the day, before we did any sort of radiography, if somebody complained about something, oftentimes, especially for GI problems, back in the day, doctors would usually just take their patient's word on it and be like, okay, we're going to have to go in there and explore and see what's up. So you would have to do exploratory surgery to see exactly why the patient was complaining of whatever, if they had pain or if they had if they felt abnormal masses or growths. But now with radio imaging, you can see stuff before you go in and actually do something more interventional, right? So it's a really good way to be able to visualize what might be causing an underlying issue with the patient. And then you can use it, of course, to diagnose. You can look for things, you can find tumors, for example, especially if you're using CT scan or MRI. MRI in particular is really, really good for seeing like soft tissues. So you can see, like, for example, even torn ligaments and stuff like that. MRI is going to be one of the best ways to visualize things like that. And then, of course, now with like more modern radio imaging, we can use things like functional MRI. And you can see different parts of the brain light up depending on what, if those portions are being activated or not. So super, super cool stuff, especially nowadays for like cognitive psych and neuroimaging for all sorts of cool things. Now, before we get into all that, we're going to talk a little bit more about EEG, or electroencephalography, which not really related to neuroimaging per se, but it's a way that you can measure the electrical activity of your brain. And it definitely has lots of benefits, and we still use EEG quite a lot today, especially if a patient has, for example, epilepsy or something like that. EEG is a very useful tool to be able to figure out where the epilepsy is originating from and what kind of things you might see on the EEG chart. Then there's transcranial magnetic stimulation, which is also really freaking cool. Literally going in there with magnets to be able to stimulate certain parts of the brain. Can also be used to treat certain types of neurological and psychiatric conditions. So transcranial stimulation has, there's some really interesting stories about people with like Asperger's getting transcranial stimulation in certain regions of the brain that can help to maybe make them a little bit more empathetic towards other individuals. You can also use it to treat things like depression, major depressive disorder. You can use transcranial stimulation to try to treat depression, for example. So really, really cool technology. Yes?
Is this like the low frequency ultrasound?
Oh gosh. No, because this is magnets.
Okay.
Whereas ultrasound is using like sonar, basically.
Okay.
It's like sonar type technology. So you said low frequency ultrasound?
Yeah. So I heard it could be used for some of the same stuff, like depression and stuff like that.
Low intensity. I'd have to look more into that. I don't know much about low frequency ultrasound, but I'll leave that in my browser so I can look up more stuff. If it's pertinent to this class, I'll include it. So let's watch a little video on transcranial magnetic stimulation. Super cool technique.
Transcranial magnetic stimulation, or TMS, has been approved by the Food and Drug Administration for the treatment of depression since 2013. A large number of studies show that most people get at least 50% better with TMS treatment, and about 30% have their depression go into remission. Let's take a look at how TMS works. In TMS, a wand is placed over the forward part of the head. What does this wand do? An electrical current moves through the ring of the wand, which creates a magnetic pulse that is pointed down at the head. The magnetic pulse passes through the skin and the skull and enters the brain. Although this is not painful, you might feel a slight tingle. This generates an electrical current in the brain, which is also painless. The magnetic pulses only stimulate a small part of the brain that is near the TMS wand, but the brain cells stimulated there can activate other areas of the brain, including areas on the prefrontal cortex and the anterior cingulate cortex. These two regions of the brain are very important in mood regulation. Prior to receiving TMS with us, you will get an MRI of the brain. A few days after the MRI, you will come to the TMS treatment room at our clinic. Using information from your MRI, the TMS machine will be programmed so that a robotic arm holding the TMS wand is set in the right position to deliver individualized TMS treatment while you lay down on a comfortable table.
So anyways, that's transcranial magnetic stimulation. Again, you can use that to treat different types of neurological or psychiatric conditions. Now, let's talk about EEGs. So this is electroencephalography. And nowadays, when we're doing EEGs, for the most part, we're doing EEGs to assess for like seizures, epilepsy, things like that. There's other indications as well. I mean, you can use EEGs for all sorts of things. You can use them for like concussions. You can use them for anything else that's neurological as well. Back in the day when EEGs were first developed by Hans Berger, German, I think he was a physiologist if I remember correctly. But anyways, he was using it for like tumor diagnosis and things like that. So you can use EEGs to be able to try to find where like maybe potential like tumor growths might be in the brain. But nowadays, of course, with CT scans, MRIs, like to look for a tumor, those are going to be the best options for you. But EEGs are still very useful. When I was doing my neurology rotations, one of my preceptors, she likes to describe epilepsy, for example, as some of your neurons just start to get a little bit too excited, and they start to like have a little bit of a party, right? And then the party is contagious, and then all the neurons in the brain start partying, right? And so you can see where the seizure starts sometimes using EEG. So you can see like the focus, the foci. Sometimes that could be because maybe the person had like trauma, right? They might have had like a traumatic brain injury. Maybe they have some scar tissue there, and that might be what is causing the seizures or the epilepsy. Or it could be a tumor, right? So tumors can actually manifest through seizures. So sometimes a person doesn't even know they have a tumor until they start getting seizures. And then they go get their EEG done, then they get neuroimaging, and all of a sudden, wow, you see that the person actually had a tumor in the brain. I'm going to skip these videos over here. It basically just talks about what it looks like when you're getting an EEG. These videos are kind of cool because it shows like a pediatric patient and the whole process of going in, getting the cap placed, getting all the conduction gel put in place as well. So that's one thing that you have to do when you get an EEG done is you have to put in conducting gel, right? So you can actually read the electrical signals. Now the reason being is because when you're doing an EEG, you're having to deal with all the other tissues that are in the way, right? So you have CSF, right? Then you have your meninges, then you have bone, then you have muscle, connective tissue, you have hair, right? All those things are going to interfere with you being able to read an EEG. And so you have to use a lot of conducting gel to be able to try to maximize your reads. But anyways, with seizures, that's going to be one of the major things that you are going to be using an EEG for, is to be able to look at different types of seizures. You can see different types of spikes that take place when a patient is seizing, when they have seizure onset. You can kind of see it on this image over here, where you have normal, and then all of a sudden the seizure begins, and then you can see the postictal, the preictal phase of the seizure. Preictal is the lead up to the seizure. We also call this like prodromal period. And the prodrome before a seizure can last a while, can even last a few days before you actually have a seizure. Usually that prodromal period, the preictal period, can be characterized by like changes in mood. The person might be feeling kind of depressed, maybe feeling a little lethargic. And then all of a sudden, before they actually get the seizure, they'll have a preictal period where they might start having like visual hallucinations, auras. They might taste something funny in their mouth. And then all of a sudden they have the ictal period. And when you say ictal, that means when the seizure is actually taking place. And then after the seizure is done, depending on what kind of seizure it is, like for example, grand mal seizures, they're going to have a pretty significant postictal period. People with grand mal seizures, after they have a seizure, for example, like maybe early in the day, for the rest of the day, they're just super lethargic, super tired and out of it, because basically their brain like used up all of the fuel, right? Used up all the energy, and now it's exhausted. So anyways, these are things that you could see on an EEG. So Dr. Hans Berger is the man who basically pioneered this whole technique. And again, he was using it to diagnose all sorts of things, tumors, strokes, other types of disorders as well. But again, nowadays, tumors, strokes, better to look at those using CT type technology. You can do a lot better neuroimaging. So what are you looking for on an EEG? You're looking for frequency, And for this one, you are looking for amplitude, right? This is not like when you're recording an actual neuron and you see just like those individual spikes, right? That you're only looking for frequency. But for an EEG, you are looking for changes in amplitude. Now, what is frequency? Frequency is basically how fast the waves occur, right? So the shorter the wavelength, the greater the frequency. Versus the amplitude is the height of the actual waves, right? So the higher the wave, the greater the amplitude. What are things that can affect your EEG? Lots of stuff. Even just movement can affect your EEG. So just like moving your head and neck, right? Because how do you move, right? You're sending depolarization. It's neuronal firing that allows you to move your skeletal muscle. So whenever you do move, taking an EEG, that's going to show up as artifact. What else? Eye movement. So whenever you keep your eyes open or close your eyes, or when you blink, you'll see like a little tiny blip showing up on the EEG from blinking. Concentration levels, especially if you're in like sleep mode or if you're in meditative states, right? You can get different types of EEG readings. So like, for example, delta waves are going to be something you see when you're in deep sleep. If you're in REM sleep, you're going to see a predominance of delta waves. If you're awake and you're concentrating on a problem, you might be in beta waves, because beta waves are going to be when you're awake, alert, your eyes are open. If you sit down and you're like closing your eyes and maybe you're like meditating a little bit, then that's going to throw you into alpha waves. So different types of levels of concentration and arousal states are going to affect your EEG. If you're stressed out, we're going to talk about stress a little bit later. That can affect your EEG. Drugs, of course, can affect your EEG. If you're on like, if you're doing rails of cocaine and you're, completely like twacked out, or if you're doing meth, or if you're doing opioids, right? Any sort of drug that has a psychological effect, neurological effect, you're going to see that on EEG. And then schizophrenia and psychiatric conditions can affect your EEG as well. We're going to talk about that in a sec. And of course, epilepsy, right? And so if you were to do an EEG in the clinic, you're going to be using a cap, and it has multiple different areas, right, that you are going to be getting your sensors, to be able to detect your different brain waves. And so here are your different brain waves. We're going to go through all of these, but alpha, that's going to be what you see, especially when you close your eyes and you're kind of maybe like relaxed a little bit. Beta is going to be when you have your eyes open, you're concentrating, maybe you're focusing on a problem. Delta is going to be another big one that we're going to talk about. That's when you're like in deep sleep. And then you also see like gamma and theta waves as well under different circumstances, which we'll talk about later. But alpha and beta are definitely some of the more predominant ones when you're awake and alert. But just note that all of these waves are all happening simultaneously, right? But depending on the circumstance, one type of wave, EEG read, will be taking over. Like for example, you're always going to have alpha waves, even if your eyes are open. But when your eyes are open, beta waves are basically going to take over. Okay, so then you get a preponderance of those types of waves, but they're always happening all simultaneously. So again, alpha waves, the frequency is going to be anywhere between 18 to 13 hertz, and then the amplitude is going to be about 30 to 50. And this is what you get when your eyes are closed. So when your eyes are closed, the frequency becomes a little slower, and the amplitude, however, gets a little bit higher. So you get a bigger amplitude when of your EEG read when your eyes are closed. This would be a situation where maybe you're calm, you're relaxed, maybe you're just kind of like in a meditative type state. Then the moment you open your eyes, then the amplitude diminishes pretty significantly. It's going to go a little bit lower in amplitude, and then the frequency is going to change as well. It becomes a little bit higher frequency with beta waves. So here's your, sorry about that. So beta waves, that's going to be something where you're going to see more when you're awake, alert. Maybe you're like solving a problem, for example. If you're doing an EEG read right now, and if you're trying to like figure out like a math problem, for example, then your beta waves are going to be the ones taking over. Delta waves, the moment you go into deep sleep, delta waves are what basically take over, as well as stage 3 sleep. But you especially see this in REM sleep, What does REM stand for? And I'm not talking about the band. Yeah, rapid eye movement. So when you're in rapid eye movement, that's when you're consolidating memories, right? Whatever you learn throughout the day, that's when your brain really is consolidating into that into like solid memory. It's also the stage of sleep when your microglia, for example, are clearing things out of the brain, right? Where you're clearing out misfolded proteins like beta amyloid plaques, tau proteins, things like that. So that's why sleep is super important, especially getting into delta waves. And you're not going to see this with conscious adults, but you are seeing this in infants that are still developing. What do infants like to do? If you guys ever been around like little tiny kids. They like to cry sometimes, but they also sleep a lot, right? Babies are sleeping a lot, especially as they're developing from an early age. And so the brains of infants that are still developing are going to be quite often in a delta state. Theta waves, this is something you also see in kids, right? When kids are awake. But then if you see it in an adult, that means the adult is probably frustrated. Okay, so maybe it's like us being frustrated, like kids perhaps. So you also see this in sleep as well, right? You see delta waves and theta waves in sleep, and then gamma waves. Gamma waves are kind of interesting. If you are like a meditator and you're really good at meditation, you're going to throw yourself into gamma waves more so than other individuals. You also see this go away during anesthesia. And gamma waves are not seen in schizophrenic individuals. Okay, so why? I have no idea, but you don't see it in schizophrenia. I would imagine. I don't think that's really going to affect your EEG, unless you're getting a stroke or something like that. But yeah, that's going to be more so with your heart. Let me look it up real quick. Can hypertension affect your EEG? Let's see what Dr. Google says here. Our studies have shown that increased BP was associated with increased cerebral electrical activity. All right, so I guess it can affect your EEG read. There's a study that was published. It's here in the NCBI. You can find this article. It's called Investigation of EEG Activity Compared with Mean Arterial Blood Pressure in Extremely Preterm Infants. So if you really want to read that article, feel free to do so. So I guess it does affect the EEG, but I don't know. Who knows how significant the effects are? I'm not sure. But anyways, these are all the different types of things that you might see affecting your EEGs, right? It could be, for example, seizures, of course, are going to affect your EEGs. Could be drugs, right? If you're overdosing on drugs. So lots of different things can affect your EEG. I just put this in here because it came from a pretty interesting study from back in 2008. I am not going to test you on any of this stuff, okay? This is just kind of to give you a feel for what might affect your EEG. So here's your EEG on the left. You can actually use computerized enhanced imaging to be able to see what areas of the brain were more affected than others. For example, in this EEG read on this kid, you can see parts of like the prefrontal cortex was lighting up. So the darker red it is, the more electrical activity. And so this individual is blinking. And so part of the prefrontal cortex might have been associated with that, especially with the muscles around the face might be triggering the EEG to increase the reception of electrical signals. Eye movement can also affect the EEG. And here's your transcranial magnetic stimulation on that image. So with this cap over here, you can measure event-related potential. That's the fancy term for what you're looking for on the EEGs. All those different waves and signals. Those are event-related potentials. Again, different areas are going to have more activity depending on what's going on, right? If you're like moving your eyes, moving muscles, or if you're having a seizure, you can see different areas light up more so than others. And again, here's your transcranial magnetic stimulation. Now, any questions on EEGs before we start talking about more like radiology type topics? All right, cool. EEG is pretty straightforward. I was debating whether or not I wanted to bring in the EEG machine, because I have an EEG machine in one of my labs for MedPhys. I might still do that for this class. I'm still debating on that. We just have a lot of material to cover, so setting up the EEG machine might take a little bit of time. So I'll still consider maybe doing that at some point this semester. It's kind of fun. It's fun to see how what your brain waves look like. All right. Here are other types of radiological approaches. You can use ultrasound, which we're not going to talk about in this class. Generally speaking, you don't do like ultrasound to the brain, right? You're mostly going to be doing X-ray, CT, and MRI. You can also do nuclear medicine, so like PET scanning, for example. That's going to be using nuclear medicine. Like you can use radioisotopes of like glucose, for example, to see if you have increased glucose uptake in a certain part of the brain, which would indicate greater activity in that area of the brain. You can also use fluoroscopy, but that's for other things. Fluoroscopy, that would be like barium, swallow. I think that would be considered fluoroscopy. Anyways, that's beyond this class. Let's focus on the brain. So let's talk about CT scan versus X-ray. So again, CT's computerized tomography, that is basically glorified X-ray, right? It's going to be X-rays, but you're getting a 360 panoramic view of the entire thing that you're trying to scan. So you get a much more in-depth image of what you're looking for. X-ray, though, it's useful for especially like orthopedics, right? If somebody breaks a bone. You can also use X-ray coupled with things like fluoroscopy, where you're injecting like dyes, for example, into different parts of the body. So you can see, like, for example, like angiograms, like coronary vessels and stuff like that. So you could couple X-ray with those types of techniques as well. But CT is going to be really, really useful to get a better image. PET scan, that's positron emission tomography. And again, that's using radio-labeled molecules. It could be dopamine. It could be things like glucose, for example. And you can see which parts of the brain might be affected. With dopamine, you can see areas of the basal ganglia to see if the basal ganglia is, operating the way it should be, right? If it's not, maybe the person has Parkinson's disease, and you would be able to detect that on a PET scan. And so here is your CT scanner, basically just a large tube, and the patient goes in, laying on their little bed, and then you can get a nice image of whatever it is that you're trying to scan. So in this case, this is a CT of the head, where you can see all sorts of different structures. And with CT scans, you can detect things like tumors, right? In this image over here, you can see a tumor right over here. You can see even like areas of metastasis for the tumor. So CT scan can be very beneficial for that as well. And then here's your PET scan on the bottom right. So this is where they were injecting the individual with like a radioisotope of dopamine to see which part of the brain was getting affected. So over here, this is an individual with Parkinson's disease. In a healthy individual, you would see regions of the basal ganglia lighting up with those radioisotopes of dopamine. If you had Parkinson's disease, your basal ganglia is going to be affected, right? And so you would be able to see that on a PET scan. And again, here's your a couple examples of CT scans. With this CT scan, you see that there's a little tiny nodule here. So this individual actually has hydrocephalus. And then down over here, this is an MRI. So you can see the difference in the imaging of CT scan versus MRI. With an MRI, you get a much clearer and more defined image, right? The downside of MRI is that it's extremely expensive. It's way more expensive than a CT scan. I don't know the exact figure, but it's in the thousands. So just one MRI study can be very, very pricey. The machines are expensive, and so I think when they do the pricing, I think they factor in the cost of the machines, right? The upkeep too. Oh my God, it's insane. The CT scanner here, we have to get because it's actually a veterinarian CT scanner, because you're scanning like small animals and stuff like that, and so we have to get this guy. He's a veterinarian, and he's the guy that's certified to do like upkeep and servicing on these CT machines. He comes like once a year, and just for like a 1 hour consultation, basically, he gets $20,000. GCU pays this dude $20,000. When I found out about how much he makes on just one measly visit, I couldn't believe it. I was like, dude, I went into the wrong field. Like, this is insane. Absolutely insane. And so just like the upkeep, the maintenance, and just to get the machine serviced, it's very expensive. And so that all contributes to the cost of, the interventions and things like that. So, all right, so here's your CT scanner. Again, we have basically the same thing here, except it's much more tiny, right? It would actually fit on this desktop over here. So super cool. Let's watch a little video on CT scanning.
A CT scan is an X-ray procedure that creates cross-sectional images with the help of computer processing. CT images are more detailed than conventional X-ray images and can reveal bones as well as soft tissue and organs. A conventional X-ray uses a fixed tube that sends X-rays in only one direction, while a CT scanner uses a motorized X-ray source that shoots narrow beams of X-rays as it rotates around the patient. There are special digital X-ray detectors located directly opposite the X-ray source. As the X-ray passes through the patient, they are picked up by the detectors and transmitted to a computer. Image slices can either be displayed individually in two-dimensional form or stacked together to generate a three-dimensional image that can reveal abnormal structures or help the physician plan and monitor treatments.
So yeah, it's super cool. You can also use CT scanners to create a file. And I haven't learned how to do this myself. I want to learn how to do it on this machine over here. But you can actually create a file that you can thereby 3D print. So you can literally look at a structure and then you can actually 3D print it. So super, super cool technology. So with X-rays, these are all the, with CT scan, these are all the different types of conditions that you can look for. Right? You can look for things that's going on in the central nervous system. Like you can look for tumors, for example. You can want to use this if you have a patient with a stroke, right? This is like one of the very first things you want to do with a patient that has a stroke. You want to send them off to get a CT scan. Why? Why do you want to do that with a stroke patient? Do you guys remember?
See if it's ischemic or hemorrhagic.
Yeah, exactly. Beautiful. So if it's a hemorrhagic stroke, you do not want to give that person any sort of like clot busters or anything like that, right? Whereas if it's ischemic, then you can proceed with like TPA to treat that stroke. But there's all sorts of different indications for CT scan. It's not just neurological imaging. So good stuff. So here's an example of CT scan. This is on a young, healthy individual with no like cognitive abnormalities. And now this is the brain of a really old individual, 87 years old. And as we get older, our brains just naturally atrophy, right? Some people have more severe atrophy, right? That especially if they have Alzheimer's and Parkinson's. And if you have neurodegenerative disorders like Huntington's as well, you would see some significant atrophy. But even just normal, senile, senility, basically, that's, going to involve atrophy the older you get. And what do you see on a CT scan? The ventricles are going to be widened on a CT scan. So you're going to see an increased size in the ventricles. Then you'll also see atrophy of the gyri. The sulci are going to also be enlarged. Right, so these are all things that you could be able to see on a CT scan. Here's an example of what you could do like a 3D rendering image of an individual with like a skull fracture, as an example. So for this individual, like you could, you do the CT scan and then you can have the option of turning that into a 3D image. So super cool. You can also do this for angiography, right? If you want to study or look at different blood vessels in the brain or the rest of the body. You can look for example, occlusions in a vessel. You could look for aneurysms. You could maybe even be able to detect berry aneurysms in the circle Willis, as an example. So you can see a lot with a CT scan. So here's your PET scanning. This is your positron emission tomography. Again, this is using radioactive isotopes and radio tracers. And so for this example, this is this person, they're using glucose to be able to see if they have reduced or heightened activity of the brain. So with Alzheimer's, this individual has much less activity, right? That's going to be one of the hallmarks of Alzheimer's, right? You have reduced cognitive activity. So again, brain activity levels, this is based on glucose uptake. Red is going to be indicating lots of glucose uptake. Blue is lower. This is a person who's manic on the right side. So when a person has mania, what's a condition that causes this? You guys know? Yeah, bipolar disorder. So bipolar 1 especially. Bipolar 1 is going to be characterized by 7 days, at least 7 days of a manic episode. Bipolar 2, it's submanic. So hypomania, and it's characterized by 4 days of a manic episode. So it's not as severe. Bipolar 1 is by far much more severe. But if you're manic, your brain is going to be all over the place. It's going to be hyperactive. People with mania, if you have bipolar 1 and you're going through a manic episode, you're going to be sometimes up all night. You're not going to be sleeping. You're going to have flight of ideas. You're going to have delusions of grandeur. A lot of people with bipolar 1, they'll be like, oh my God, I'm going to start this new business. I'm going to do all this really cool stuff. And then they spend all their life savings on their plans. And then they lose all their money. Sometimes they'll go on gambling sprees. They'll like go gamble all their money away. Sometimes they'll go on sex binges and they'll literally like sleep with anything that moves. And I know someone personally that's happened to. Like, there's this guy I know, and his wife has bipolar 1, and she basically did that. She cheated on him for like weeks, and he was just having like nervous breakdowns and stuff. They've got like 3 kids together. He is a stronger man than I. When she came back to, he forgave her, basically, like brought her back into his life. Kind of crazy. But anyways, so that's mania. When someone's manic, they're like on top of the world. They feel like they're on top of the world until they're not. And then like their life collapses and falls apart. People with bipolar 1 generally wind up in the hospital, right? That they usually get hospitalized. And they have to, because otherwise they're going to destroy their lives. They'll destroy their own lives, or they'll sometimes destroy other people's lives too, because they can tend to be violent as well. People are always concerned about schizophrenics, but it's usually bipolar that tends to be a little bit more violent. The most violent patients I've ever seen when I was working in the hospital were like super manic bipolar patients. And it's a little scary because they don't like to take their drugs. Why don't they like to take their drugs? Because being manic is awesome. They feel like they're on top of the world, right? Like they're awesome. And then when they take their drugs, then they feel boring and lame and normal, right? So they don't like taking psychiatric drugs in general. Will they feel depressed if they take like?
Do they feel depressed based on what they feel when they're not on the medication?
Well, bipolar is a type of mood disorder that involves depression. So bipolar depression, right? After mania, then they go into a really deep depression after their manic episode. But so when they take mood stabilizers like lamotrigine is a really good drug for bipolar patients. Lithium plus lamotrigine is a really good combination. Lamotrigine is a anti-epileptic drug, but it can also be a mood stabilizer for these types of patients. So it can help stabilize things. So they won't have the extreme highs, but they also won't have those extreme lows as well. So it gives a little bit more of a balance. But psychiatric drugs in general tend to be, like even like antipsychotics, like they can be kind of, you can make you lethargic. Even anti-epileptic drugs can make you pretty lethargic too. So not easy to treat. Let's put it that way. Bipolar is especially difficult to treat, just because nobody wants to take their meds, All right, so here's your MRIs. Again, MRIs are going to be giving you a much, much clearer image on things like soft tissues. Anything that has lots of water in it, anything that has lots of fat in it, those tissues are really going to shine when you use an MRI. So you can use this for if you're looking for infarct, ischemia. You can use this for vascular changes, hemorrhage, infections, all sorts of stuff. Lots of indications. These are really fun videos. I love these videos that show like people's eyes moving with an MRI. Super, super cool. Now this is somebody rubbing their eye. This is what happens whenever like you have an itch or whatever and you're like rubbing your eye. Look how far back the highlight goes into its orbit. Pretty wild. So super, super interesting. I love these videos. They're cool. There's a couple other ones I want to show you too. Jeez Louise. That's an even better image. So you can see your lateral rectus, your medial rectus, and all that. Cranial nerve 2. Super, super cool. All right, let's see what this last one is. I can't remember off the top of my head. Oh. So this is using functional MRI. So with a functional MRI, you can see different areas that light up. And with the areas that light up, that indicates, of course, heightened activity in those regions. Functional MRI nowadays is being used for all sorts of different types of research. Really, really cool stuff. Again, extremely expensive. So, I mean, I wish we had that type of capacity here on this campus, but we don't, unfortunately. But maybe someday, who knows? So I'm not going to play this video over here, but that will give you some more insight into how the actual MRI works, so you can watch that on your own time. And it basically has to do with magnetic fields and the spin, right, of different different types of atoms and whatnot. So when you run an MRI machine, when you turn it on, you turn on a magnetic field. So different types of things are going to spin at different frequencies, different energies. And so that's how you're able to detect soft tissues, for example, anything that's high in water and high in fat. And then here's your like a tractogram, also referred to as diffusion tensor imaging. I will play this one because I think it's super cool. So I'll play a little bit of it, not the whole video.
Neuroscience Methods 101. Today, we will be discussing diffusion tensor imaging, or DTI. The white matter tissue of the brain contains axons which form the connections between neurons. Making a map of these white matter fibers allows for understanding which areas in the brain are strongly connected. Consequently, this gives an idea from where to where information is flowing in
You see the corpus callosum right there in the middle, that pink spot. It's super cool.
However, it is very difficult to actually see the different connections and along which direction different axonal bundles are going. For that, we need DTI. As we already have seen in our introductory video on MRI, an MRI scanner is great at picking up hydrogen atoms. So in a structural MRI scan, tissues with more or less water content can be distinguished. Importantly, hydrogen atoms don't stand still, but are constantly moving around. But they cannot just go anywhere they want. For example, within white matter, hydrogen atoms mostly move along the direction of the axon, similar to water moving through a straw. Now, let's take a more detailed look at how DTI can detect the differences in hydrogen atom movement in the brain. In the MRI scanner, hydrogen protons align themselves to the magnetic field and spin around their own axis, all in the same phase. First, let's take a look at an example where hydrogen atoms do not move. When performing a DTI scan, a gradient is added to the magnetic field. As a consequence, the synchronized spinning of the protons is disrupted, and depending on the magnetic field, some protons spin faster and some others spin slower. If we afterwards apply another magnetic field gradient that is exactly opposite to the first and is applied for the exact same amount of time, the effect of the first gradient would just be nullified. In other words, at the end of the application of the second gradient, all protons would be spinning in the same phase again, just as they did in the beginning. Now, this example is only true if hydrogen atoms wouldn't move, but of course in reality, they do move. In that situation, just as in the first example, the first gradient would cause the protons to spin differently from each other. But because the protons are moving, the second gradient will not have the same effect as the first.
All right, I'm going to, for the interest of time, stop that right there. If you want to watch the rest of that video on your own time, feel free to do so. So that allows you to visualize the different tracts. So it can cause you can get nice little tractogram type images. So super, super cool stuff. This is how the MRI works. Whenever you're doing MRI, even functional MRI studies, especially with the functional MRI study, you want to see like what's going on in different parts of the brain. For research purposes, the participants can wear goggles, right, that help them to like maybe solve problems or maybe like light up in different ways or whatever. And so it can help like, see like cognitive processing of visual stimulus and stuff like that. So very, very useful tool for research purposes. Again, you're using magnets to be able to basically stimulate your water molecules as well as protons and things like that. So here are a couple examples of MRI. So this is the opening slide that I showed you guys. So that's a color enhanced image of the head and neck. Here you can actually see the conus medullaris. You can see the parts of the cauda equina that come off of the very end of the spinal cord. And this is what, L5, L4, L3, L2, and you can see that's where the conus medullaris sort of ends, right? It's around L1, L2, right? So if you're going to do a lumbar puncture, you would want to either go through L3, L4, or L4, L5, right? So you can avoid striking the spinal cord. So here's another image of the spinal cord. Again, you can see that cauda equina, conus medullaris, etc. Now, this is not from your textbook. This is another anatomy book that I have that had some really beautiful images that give you examples of what you could see with MRI. So super cool. You can see lots and lots of detail using MRI for neuroimaging. Got your corpus callosum again, commissures, all your tracts and all of that. So moving on. Now, with functional MRI, this is where you see different areas lighting up. So why would you use this? For research purposes, you could like maybe administer stimuli to a patient and see which part of the brain corresponds with the processing of that stimuli. So for this case, this is painful stimulus. They were actually subjecting the participant to different painful stimuli, and you can see different parts of the brain that light up as a consequence. So super neat. You can also use it for tumor detection. So this is a really nasty, massive meningioma. You can also use it for infections. So this is an example of encephalitis, which is generally going to be viral, not to be confused with meningitis, because meningitis only affects the meninges, whereas encephalitis actually affects the brain. And one of the most common organisms, or not organism, one of the most common viruses is going to be HSV2, so herpes simplex virus. And then here's your diffusion tensor imaging. Again, I'm not going to play this video because we already watched one, but it allows you to really see all of your major tracts. So it's also referred to as tractogram. So it's called MRI tractogram. And you can see where all those different regions go down. So here you can see the corpus callosum right here in the middle. And you can see all the different tracts coming in from like your motor cortex, somato, sensory cortex, and all that stuff. So super, super cool. And if you want to get even more granular information, this is still an fMRI. We're going to talk about magnetograms in a second, but still fMRI where you're seeing areas lighting up due to different types of stimuli. And then here's another image of fMRI. You can see different areas lighting up depending on the stimulus. And again, it's going to be based on metabolic activity of those regions, right? The more metabolically active a region, the more it's going to light up. Now with magnetoencephalography, this is even more detailed. You get a very detailed image. And it's going to be very similar to MRI. It's going to be recording magnetic fields, but it's going to be a little bit more detailed. It's more sensitive. So the magnetometers are going to be more sensitive. So here's an example of what that MEG or magnetoencephalography looks like. So these would be the type of imaging that you would see. And it's really good for time, right? So you get higher resolution in terms of time. So greater temporal resolution. That's one of the benefits of using this instead of an fMRI. fMRI you're going to get less temporal resolution. So milliseconds. It's going to be in milliseconds. I'm not exactly sure what the time measurements for MRI are, but it's going to be a little bit greater than that. And so for this example, they were doing a study on having this person move. And so before the person actually moved, you had the anticipatory activity. So remember, we had like your motor cortex, right? You got your precentral gyrus. You also have like the motor association areas, right? Those are all like the anticipatory regions of the brain that are kind of like starting to formulate the idea of movement until you actually do the actual movement, right? And so before you move, you're going to have some anticipation before that, and then you can see the areas of the brain light up corresponding to the actual movement. So those are why you would use one of those types of studies. MEG would be good for that type of study. And so here's a summary why you would use different types of neuroimaging for perception, language, cognitive type studies, things like that. So it's really huge in cognitive neuroscience. We're going to get into cognitive neuroscience at the very end of the semester, but today we are at the end of lecture. So we're going to do these quizzes questions next time we meet next week, and then get into embryologic development. Okay? Any questions? Cool. All right, guys. Well, it's good to see you guys again in person. Have a wonderful weekend. See you next Tuesday.
Do you go over things the lecture before, or do you have
So my exams are going to be a lot of the stuff that you see in the quizzes. So the topics that we see that I put in the quizzes, those are going to be the types of things you're going to see on the exam, right? So of course I'm going to reword them, I'm going to restructure them so it's not the same type of question, but the concepts are going to be similar. So that's usually how I structure my exams and my quizzes. I do it both so I can help you get a better feel for what might actually show up on the test. And like I said before, I'm never going to just out of left field put something on the exam that I haven't talked about in class. If I haven't talked about it in class, it's not going to be in the exam, okay? Or if it's not in the quizzes, it's not going to be in the exam. Sound good? All right. Awesome, guys. Well, have a wonderful afternoon, and we'll see you next week.